Good morning. My name is Udbhavram, and today I will be presenting my research on the quantitative analysis of gel electrophoresis images. So before we dive in, what exactly is gel electrophoresis? Well, it's a very widely used technique that is used to separate protein fragments according to their size. Here we can see a typical gel electrophoresis with a ladder, a negative end, a positive end, and the gel in the middle. We can also see that we're going to use 3 samples, a large, medium, and a small sample. Now, when we place the large sample in the well and allow the induced electric current to act on it, we observe that it moves from the negative end to the positive end. This is because these protein fragments are negatively charged and therefore would like to move to the positive end. However, we notice that it cannot move all the way to the positive end and stays relatively close to the negative end. This is because the gel itself provides resistance based on the size of these samples. And we can see that when we add a medium sample or a small sample. Now, another thing that we notice is we can't quantitatively analyze these samples other than saying that they're large, medium, and small. Well, the way that we quantitatively analyze these are with ladders. These ladders provide a base for us and a reference point with which we can compare our samples to give them specific numbers. So in this case, we can draw a line straight down and we can say that these points correlate to those points on the ladder. And in doing so, provide a quantitative analysis of the gel. Now, the problem is not all gels are perfect and not all gels will look like perfect little rectangles. Often in our lab, with the origins of life scans, we're dealing with nucleotides that are very small or come with other nucleotide lengths in that same sample. And what this results in is a visible smearing across the gel. With these types of scans, it becomes very difficult with ill-defined columns to identify using human vision the exact points at which we have our concentrations of interest. On the other hand, with an ideal scan, as we've just seen on the other slide, and you can see on the right side of your screen, you have multiple well-defined columns with specific intensities that we can correlate to specific values on our ladders. The overarching issue is that there are major limitations with human vision, human precision, and the manual tools that we use. So we believe that the solution is cloud-based analysis software. Here we'll be walking you through the software and the application. On the first page of the application, we can see that we can select an image to upload. Once pressed, this will now take you to your photo gallery. You can now choose from any number of photos that you've just taken on your phone or that you've uploaded or downloaded from a different device. The next step, once you've selected the image, is to select the ladder qualities. So telling the system how many ladders are there and what are the qualities of those ladders. The final step is to touch the ladder points on the image to give the system a chance to figure out spatially where these ladders are. Now, what happens when we click the send button? Well, the app sends all of this data to the server. So we send 3 types of data mainly. The first type is metadata, which includes a name, an ID, an email, and anything else that isn't involved in the actual analysis process itself. Now, the second type of data is an image with the ladder coordinates, and this is arguably the most important part of the data. This is because we send the image as an encoded string to the server, along with the touch points that the user has identified as being the ladders. And the third type of data is the ladder length values. And this is what you've seen in those blue boxes where we're identifying how many ladders there are and what are the qualities of those ladders. Now, once the server receives all this, it begins the cloud-based analysis process that we'll now go into using this data that's provided by the app. And then what it returns is this returned analyzed data along with an annotated image, as well as an email to the user with the raw data. Now, once this image reaches the server, what exactly do we do? Well, the first objective is to see the image, is to visualize the image from the computer's eyes. So the first part is we take the original image and we have it run through a dynamic binarization process. All this does is convert the image to a pure black or white pixel density. This allows us to have a clearer view of the image and to analyze it more effectively. The next step is running it through a k-means clustering algorithm. All this does is cluster algorithms based on certain sizes or dimensions or shapes and groups them into like pixels. The last step is to identify and then assign these ladder values and these column values, essentially, all across the image in terms of our columns of interest, as well as our ladders. Now, the next step is, how do we read the image? Well, as humans, we often turn to tools such as rulers to help us identify real-life units such as centimeters to the units on the gel, such as base pairs or nucleotide lengths. Well, with computers, it's not as easy because we don't have a ubiquitous tool such as a ruler, which stays constant over all types of images, pixel densities, aspect ratios, and device parameters. So what we do is use pixels. And these pixels stay standard across all images and provide us with a standardized unit of measurement, which we can then correlate to our real-life values, in this case base pairs, on our image and on our ladder more specifically. So the first step in the analysis process is to now plot the intensities of our ladders as well as the pixels in the y dimension. And this allows us to get a specific look at what exactly the ladder points correlate to on the image. So we now have a spatial recognition from the user input and the column identification algorithm, as well as the user provided and user defined ladder values. So we're now pulling together a spatial awareness as well as a real life unit of measurement for these ladders. Now, the second step is arguably the most important step, and this is fitting these values exponentially. So we use an equation, as you can see here, to fit our values that we've extracted from our ladders and correlate them to the user-defined inputs. And we can see that this fit here fits the data quite nicely and gives us a sliding scale, most importantly, between pixels on our image and real-life nucleotide units. We use the SciPy induced curve fitting algorithm to generate these curve fits. Now, the third step is making these measurements. So what exactly does it mean to make a measurement? Well, in this case, it means to identify exactly the nucleotide lengths of these specific columns. So if we take the column on the left here, for example, on the y-axis, we're providing arbitrary intensities, just as with any other scan or any other graph. But the real trick is now on the x-axis, where we're providing the number of nucleotides. And this is taken directly from our exponential fit. And through that fitting, we're able to then generate this correctly scaled x-axis with number of nucleotides. And we were able to identify these high intensity peaks on the graph, which then give us a specific x value, which we know to be our number of nucleotides. So in this case, we can see that there are high intensities detected at 12 and around 15 base pairs. And we know that these are now our values, and we can return this to the user. So you may be asking, well, how do we verify that these results are correct? Well, we do an extensive stress testing process using a Python script generation of sample data, which allows us to stress test various system components. So we can see that in this scan, for example, we have three ladders with 2 points each. And we have many more ladders, many more columns, and many more data points per column. And we can change any number of things, such as ladder qualities, the exponential fit qualities, the resolution, and even specific alterations to make sure that our program works well. And this allows us to do an accelerated beta testing process by simulating tough images, and that exposes internal shortcomings before the user can ever report them. So in summary, we know that there are limitations with the current solutions that exist, and namely reliability, vision or ocular limitations, and precision. We've developed a method of analyzing gels through a cloud-based system. We use computer vision, k-means clustering, and mathematical analysis to precisely identify the quantitative qualities of these systems. And the application and resulting data are modular and can easily be integrated with no additional infrastructure or costs, and the data provided is quite rich, meaning that this can be used for publication or, depending on the user, for various different uses. I would like to acknowledge the Laboratory for Membrane and Protein Dynamics, as well as Mcmaster University and Dr. Michael Reinstatter, Sebastian Himbert, Dr. Tectiana Loskotova, and Alex Dujardin for having enabled this research. Thank you very much.